everyone, welcome to my channel Anupma Biology Classes, and this is the sixth video in the series of Human Physiology. Today we will understand about the human muscular system, and this video is the section A of part 6. At first, we will know about the movement, levels of movement and types of movement. Here, the act of changing place or position by the entire body or by one or more of its parts is known as movement. And we discussed earlier in the first video that movement is on three levels. First one is on cellular, then organ, and then organism level. Now, on the basis of systems, there are two types. First, in case of the non-living system, induced or passive movement, and in case of the living system, autonomic or active movement. So what is the difference in the induced and autonomic movement? Induced movements are like water flows in the river or an automobile runs on the road. Here, water flows on the earth due to gravity and the automobile runs by energy of the fuel. So these movements are produced by the outside forces, while in autonomic movements which are affected by the organism themselves without external forces. Now, the Brownian movement is due to the intrinsic kinetic energy of the molecules. After that, types of movements, and movements are of two types. First one is non-muscular movements, which includes streaming of protoplasm, pseudopodial, flagellar and ciliary movements. In streaming of protoplasm found in case of the amoeba and most of the cells like leukocytes. Pseudopodial movements in amoeba, flagellar movements in hydra, sperm movements or in female genital tract has flagellar movement for the movement of eggs, and ciliary movements in cell lining of the trachea, oviduct and vas efferentia. Second is muscular movement. It is of two types. First one is locomotion and second is the movements of body parts. So in case of the locomotion, it is the movement of an animal from one place to another. Locomotion takes place by walking, creeping, hopping, running, flying and swimming. So for this type of locomotion, different type of adaptations are found in case of the animals. Like for running, cursorial adaptations; for hopping, saltatorial adaptation; for swimming, natatorial adaptation; and for the flying, volant adaptation. Locomotion is the outcome of arrangement, interactions and coordination of the nervous, muscular and the skeletal system of the body. Now the second is movements of body parts, and it is of two types. First one is the movements of the external body parts and second is the movements of internal body parts. Movements of external body parts like presence of parapodia, legs, foot, 2 feet, fins or limbs, and movement of head, trunk, mouth, eye, tentacle, etc. are also in this type of locomotion. While the movements of internal body parts are like heartbeat, peristalsis, movement of alimentary canal, movements of diaphragm, movements of genital tract, etc. After that, the movements in case of the invertebrates, and in invertebrates they have no skeleton, and at the place of this they have body cavity in which fluid present, so it's known as hydrostatic skeleton. Like in hydra has longitudinal and circular muscle process that shorten and lengthen the body. In case of earthworm, moves by the contraction of the body wall muscles aided by the setae. In leech, moves by the looping action with the help of anterior and posterior suckers, and in the starfish moves with the help of 2 feet. In the picture you can see here, the hydra shows different type of movements like dragging, climbing, looping, somersaulting, walking, floating or gliding. Here you can see the looping movement of the hydra, and in this picture you can see the 2 feet or podia. Here the starfish moves with the help of this 2 feet. Now the movements in land vertebrates, and there are three types of movement in the land vertebrate. First one is amoeboid movements, which is affected by the pseudopodia formed due to the streaming of protoplasm like macrophages and leukocyte, cytoskeleton elements, etc. Second is ciliary movements. And cilia of the cells lining of the trachea, oviduct and vociferentia propel by their coordinated movements dust particles, eggs and sperms respectively in specific directions in these organs. And the last is muscular movement, and the movements which requires the use of skeleton, joints and muscles are known as muscular movements. So, in the following account, we shall discuss all these structures as well as mechanism of muscle contraction. So, the first is muscle system. And the muscle system consists of muscle tissues which consist of muscle fibers. Muscle fibers have some properties like contractibility, electric excitability, extensibility and elasticity. And the branch of science that deals with the study of muscle is known as myology. In case of the human, there are 639 separate muscles which makes 40 to 50% of the body weight. In muscle system, there are three types of muscle. First one is the striated muscle, then a smooth muscle, and after that the cardiac muscle. So, the first one, that is the striated muscle, which is also known as voluntary muscles. Striated muscles attached with the skeleton and the function with the skeleton. So, it is known as the skeletal muscle. Now you can see the picture. This is the striated muscle magnified view, and this is also known as the voluntary muscle because their activities are under the control of organism. It causes limb movements and locomotion in vertebrates. It is multinucleated cell bonded by the sarcolemma which contains sarcoplasm and well developed sarcoplasmic reticulum. Sarco word used in case of the muscles. In the picture you can see every muscle has dark A band which has middle light H zone and light I band which is crossed through its center by the Z line. The part of muscle fibers between two Z lines is called one sarcomere, and this sarcomere is the unit of muscle fibers. Each sarcomere is bundle of two myofilaments. Primary myofilament, which is made up of protein myosin and which is thick filament, and the secondary myofilament, which is made up of protein actin, and this is thin filament. The secondary myofilament has two regulatory proteins which are tropomyosin and troponin. Now the second, that is the smooth muscle, which is also known as involuntary muscle. It occurs in internal organs and responsible for their movement, so it also termed as visceral muscle. It is spindle shaped uninucleate cell without striations, and its action is not under control of animal, so it is known as involuntary. Third is cardiac muscle. It occurs in heart wall and brings about heartbeat. The end of the cells have intercalated discs for cell-to-cell relay of the signals during heartbeat. It is cylindrical, uninucleate, striated and branched. In the picture you can see the types of muscle tissues, and these are skeletal muscle, smooth muscle and cardiac muscle. In case of the skeletal muscle, these are multinucleate, smooth muscles are uninucleate and cardiac muscles are also uninucleate. Skeletal muscles are voluntary in action, smooth muscles are involuntary in action. After that, the muscle contraction. And for the muscle contraction, there is a need of motor unit. And in every body, muscle is composed of several muscle fibers which is under control of single motor neuron, that is axon. So, a motor unit consists of a motor neuron and the group of muscle fibers innervated by its axon constitute a functional unit. Motor unit are not same in all organs. Like in finger muscle, 3 to 6 muscle fibers is present, while in case of the eyeball, 10 muscle fibers are present. In this picture you can see the motor unit. Here a motor neuron which is yellow in color, which attached to the red, that is muscle fiber, and here this motor neuron and the muscle fiber combine to form a one motor unit. An axon of a motor neuron widens to form a motor end plate at the sites where it meets the skeletal muscle fibers. This site is known as neuromuscular or myoneural junction. In the picture you can see this is the neuromuscular junction. Here the axon of motor junction combined with the muscle at a point. This point is known as neuromuscular junction, and here different type of enzymes released, and because of this enzymes the muscles activated. So this motor junction, that means neuromuscular junction, is required for the muscle contraction, and the motor unit function on all or none principle when stimulated. That is, the minimal stimulus which causes contraction causes a complete contraction. A strong stimulus will not cause a greater contraction. So the ability of nervous system to increase the strength of contraction by activating more and more of the motor neurons controlling the muscle is recruiting of motor neuron. Now, the threshold stimulus. A specific minimum strength of the nerve impulse required for exciting a muscle fiber to contract is known as threshold stimulus, and it varies from fibers to fibers in the same muscle. After that, single muscle twitch. Single muscle twitch is in a striated muscle which respond to a single stimulus like a single electric shock by the single quick contraction occur. This single isolated contraction of a muscle fiber caused by a single nerve impulse or artificial stimulus, which is called single muscle twitch. After this, the muscle fibers relaxes. Then tetanus or tetanization. The state of sustained contraction due to many quickly repeated stimuli is known as tetanus, and because of tetanus we can hold a book for a long time to read, or we can carry a cup of tea across a room without a mishap, and tetanus is necessary to maintain the posture and form of the body. After that, the muscle tones, and all relaxed skeletal muscles always remain in partial contraction as long as their nerves are intact. This sustained partial contraction is called muscle tones. And the picture you can see here, the bone is connected with the muscle with the help of tendon. So tendon is a joining point for the bones to the muscles, and in the muscle there are muscle fibers which are responsible for the muscle contraction. Now, the mechanism of muscle contraction. The mechanism of striated muscle contraction is based on sliding filament theory. According to this, the muscle contraction results from the sliding of thin myofilament past the thick myofilament rather than by shrinking of the myofilaments. This theory proposed by A. F. Huxley and R. Niedergerg and by H. E. Huxley and Jean Mansion in England in 1954 independently. So this picture shows the sliding filament theory, and in this picture you can see a structure of a one sarcomere, that is the muscle unit. In this, at the time of contraction, the thick myosin filament come in contact with thin actin filament and slide on them. This pulls the thin actin myofilament towards the middle. At this time, the two Z line comes closer and the sarcomere becomes short, and there is no change in A band. So the contracted muscle becomes short and the thick, but volume remains same. After that, in relaxation, the actin filament starts to slide over myosin filament, and because of this, every part of sarcomere comes on the original place. So this is the complete sliding filament theory, and after that, the biochemical and electrical events in the muscle contraction. Here you can see the three things. First one is stimulation, then contraction, and after that the relaxation. So the first is stimulation. In this picture you can see the stimulation of the muscle fiber with the help of motor neuron. Here the nerve impulse arrives at the neuromuscular junction, and the vesicles in the end plate of the motor axon release acetylcholine, which diffuses across the nerve muscle gap. This acetylcholine depolarizes sarcolemma and is then inactivated by acetylcholinesterase. Why? because to check overstimulation of muscle fiber. So because of this, sarcolemma returns to the resting potential in polarized state. After that, electrical depolarization changes the permeability of sarcoplasmic reticulum, causing somehow the release of calcium ions from the sarcoplasmic reticulum lumen into the cytoplasm of muscle fiber. So the calcium ion binds to troponin, making tropomyosin moves to uncover the active sites on actin, and when the actin sites activated, then the muscle contraction starts. So the second is contraction. In case of the contraction, the resting membrane potential, the outside of the sarcolemma is positively charged, while the inside of the sarcolemma is negatively charged. Here sodium ion is present in large amount in outside of the sarcolemma, while the potassium ion present in large amount in inside. And the sarcolemma is more permeable to potassium ion than sodium. When a muscle fiber is stimulated by the nerve impulse, the sarcolemma becomes more permeable to sodium ion than to potassium ion. And so because of this, sodium ion move along the concentration gradient and the electrical gradient, which makes the inside positive and outside negative of sarcolemma. This state is called depolarized, and this change is known as action potential, which is signal of the muscle contraction. So after this signal of muscle contraction, that is after the action potential, ATP joins myromyosin head that hydrolyzes ATP into ADP and inorganic phosphate, releasing energy, and the energy raises the head to high energy state. An enzyme myosin ATPase catalyzes the reaction in the presence of calcium ion and the magnesium ions. Here the ATP converted into the ADP and inorganic phosphate and energy with the help of enzyme myosin ATPase and calcium ion and magnesium ion. Then the energized head is strained and joins active site on actin, forming a cross bridge, and the head now releases ADP and the inorganic phosphate and relaxes to its low energy state and position. A new ATP molecule joins the head, detaching it from the actin myofilament. Above 4 steps are repeated many times during one contraction to draw the thin myofilaments for the inward. So in this picture you can see a relaxed muscle cell and after that a contracted muscle cell. After that, in the whole process of muscle contraction, in this picture you can see the first, second, third, fourth and fifth point. So this is the process of whole muscle contraction, and here you know, in this, after the stimulation by the motor neuron to muscle by release of acetylcholine, which is a neurotransmitter which depolarizes the postsynaptic muscle cells of the sarcolemma and set up an action potential. Because of this, calcium ion releases by the sarcolemma which binds to troponin, which makes tropomyosin to move and uncover the active sites on the actin molecule. Because of this, actin binds to myosin, and here an ATP molecule joins the active site on the myosin head. You can see the picture, and hydrolyzes this ATP to ADP and inorganic phosphate, and here an enzyme myosin ATPase catalyzes the reaction. Then the inorganic phosphate release. This is the binding of myosin to actin. After that, it the power stroke and the myosin in the lower energy form. So now the this is after the energized myosin head join an active site on actin filament, forming a cross bridge. Then myosin head releases ADP and relaxes in the low energy state. This relaxation bends the head to its original position, pulling the actin myofilaments toward the center of the sarcomere. After that, head detached from the actin filament, which new ATP binds to it. This is a repeated cycle. So in this cycle you can see at first the binding of myosin to actin, and this is the activated stage. After that the power stroke, then the myosin in the low energy level, then ADP releases. After that a new ATP binds to the head, and then unbinding of myosin and actin, and after that ATP is hydrolyzed and the cycle is start repeatedly. So After this process of the contraction, there is a process of relaxation, and in this process you can see now the calcium ions are quickly returned to the sarcoplasmic reticulum by the active transport with energy from ATP. Troponin tropomyosin complex shift back to block the active sites on the actin. The thin myofilaments return to their original resting potential, that means position, and the muscle is now relaxed. In the relaxed period, sarcolemma becomes normal or repolarized as the sodium potassium exchange permit starts working. In the case of repeated contractions, ATP is replenished by the action ADP plus phosphocreatine that forms ATP plus creatine. In case of excessive muscular activity, the muscle fibers run into oxygen debt and stop contractions due to accumulation of lactic acid. In a resting muscle, the excess lactic acid is oxidized and this enables the muscle to resume contractions. So, in above all the reactions in the muscle contraction, oxygen is used, carbon dioxide is produced, glucose is consumed, and heat is generated. Now, the muscle fatigue. The reduction in the force of contraction of a muscle after prolonged stimulations is known as muscle fatigue. A muscle gets fatigued sooner by strenuous exercise than by mild exercise. A fatigued muscle pains. Now, the Cori cycle. What is this? This is the Cori cycle between the liver and muscles. In this cycle, the lactic acid formed is carried in the liver by the blood where it is converted into glycogen. The glycogen releases glucose in the blood which is reconverted to glycogen in the muscles. This lactic acid glycogen glucose glycogen cycle is called Cori cycle. It shows the role of liver in the metabolism of muscle contraction. So in the picture you can see in the liver the sufficient oxygen present. So because of this here gluconeogenesis occurs, that means conversion of glycogen to glucose. This glucose enters into the muscles for the action and at the time of vigorous exercise if ATP is used faster than the muscle fibers can produce it aerobically. Then the muscle fibers start anaerobic respiration, that means glycolysis, and replenish ATP. This produces lactic acid as byproduct. This lactic acid accumulates in the muscle cells and diffuses out into the blood, and from the blood it enters into the liver. Here it converted into the glycogen and this process occur repeated by many times. So this is the complete Cori cycle. Here, the role of liver is the metabolism of muscle contraction. Now, the skeletal muscle fibers. Skeletal muscle fibers are of two types, red and white. So, first is red skeletal muscle fibers, and these are thin, red, and slow contracting muscle fibers. They contain heme protein pigment, that is myoglobin, and abundant mitochondria and poorly formed sarcoplasmic reticulum. Example of the red skeletal muscle fibers are extensor muscles of the back in man, and the second is white skeletal muscle fibers. These are much thicker, lighter in color and fast contracting muscle fibers. They have a low content of myoglobin, few mitochondria and a well formed sarcoplasmic reticulum. Example is eyeball muscle. In the picture you can see in the eyeball muscles are the white skeletal muscle fibers. So this is the end of video on the muscle structure and its contraction and relaxation. It means mechanism of muscle. The next video will be on the human skeletal system in the second section of part 6. For more such videos in the biology, stay tuned with Anupma biology classes, and if you clearly understand this video, like and share it and subscribe to my channel. If you have anything to ask related to biology, please write in the comment section below. Thank you for watching this video.